Romila Lemos, Assistant Professor from the Department of Forensic Biology, Institute of Forensic Science, Mumbai. Today's topic of discussion and learning is DNA fingerprinting Part 4. I am presenting this topic on behalf of Dr. Archana Mahakalkar, Ms. Deepika Bhandari and myself. We will be covering the following modules. Module 1, Electrophoresis of DNA fragments. Module 2, Southern blotting. Module 3, Hybridization of DNA fragments with the probe. Module 4, Autoradiography. Module 5, Advantages and disadvantages of DNA fingerprinting. Module 6, Case studies, and Module 7, Conclusion. DNA can be extracted from almost any human tissue. Sources of DNA found at a crime scene might include blood, semen, tissue from a deceased victim, cells in a hair follicle, and even saliva. DNA extracted from items of evidence is compared to DNA extracted from reference samples from known individuals, normally from blood or saliva. Following the DNA digestion process, the resulting DNA fragments are separated by size via electrophoresis in agarose gels. During electrophoresis, DNA which are negatively charged migrate towards the positive electrode. As DNA fragments move, their migration rate is slowed by the matrix of the agarose gel. Smaller DNA fragments move more rapidly through the pores of the gel matrix than the larger DNA fragments. The result is a continuous separation of the DNA fragments according to their size, with the smallest DNA fragments moving the greatest distance away from the origin. Agarose gel electrophoresis is the most effective way of separating DNA fragments of varying sizes ranging from 100 base pairs to 25 kilo base pairs. Agarose is isolated from the seaweed genera Gelidium and Gracilaria. During gelation, agarose polymers associate non-covalently and form a network of bundles whose pore sizes determine a gel's molecular sewing properties. The use of agarose gel electrophoresis revolutionized the separation of DNA. DNA was primarily separated using sucrose density gradient centrifugation, which only provided an approximation of the size. To separate DNA using agarose gel electrophoresis, the DNA is loaded into a precast well in the gel and a current is applied. The phosphate backbone of the DNA and RNA molecule is negatively charged. Therefore, when placed in an electric field, these DNA fragments will migrate to the positively charged anode. Because DNA has a uniform mass to charge ratio, DNA molecules are separated by size within an agarose gel in a pattern such that the distance that is travelled is inversely proportional to the log of its molecular weight. The rate of migration of a DNA molecule through the gel is determined by a number of factors. First, the size of the DNA molecule. A larger molecule takes longer time to go through the pores and hence travels slower than smaller molecules which can easily pass through the pores. The second factor is the agarose concentration. The higher the concentration of agarose, the smaller is the pore size and vice versa. The third factor is DNA conformation. A closed conformation of DNA due to its compact nature will allow easier migration than a linear structure that may face hindrance. The fourth factor is the applied voltage. The voltage applied dictates the availability of the ions and thereby affects the rate of migration. The fifth factor is the presence of ethidium bromide. Ethidium bromide or ETBr, being an intercalating agent, blocks the negative charges of DNA, which may hinder the movement of DNA molecules. The sixth factor is the type of agarose. A purified agarose will form better cross-linking and allow better migration. The seventh factor is the electrophoresis buffer. Different buffers have different ionic compositions which can in turn dictate the rate of migration. After separation, the DNA molecules can be visualized under UV light after staining with an appropriate dye. Let's have a look at the procedure of this technique. The first step is the preparation of the gel. The appropriate mass of agarose is weighed and taken into a conical flask. The concentration of agarose in a gel will depend on the size of the DNA fragments that are to be separated, with most gels ranging between 0.5% to 2%. The running buffer is then added to the agarose containing flask, taking care that the volume of the buffer should not be greater than 1/3 of the capacity of the flask. The most common gel running buffers are TAE, that is tris acetate EDTA buffer, and TBE, that is tris borate EDTA buffer. This is mixed well. After that, the agarose buffer mixture is allowed to melt. This is most commonly done by heating in a microwave, but can also be done over a Bunsen flame. After every 30 seconds intervals, it is important to remove the flask and swirl the contents to mix it well. This is repeated until the agarose has completely melted and a clear solution is formed. Next, the dye ethidium bromide or ETBr is added to a concentration of 0.5 micrograms per ml. Alternatively, the gel may also be stained after electrophoresis in a running buffer containing 0.5 micrograms per ml ETBr for 15 to 30 minutes, followed by its destaining in the running buffer for an equal length of time. It is important to note that ETBr is a suspected carcinogen and must be properly disposed of as per the institution regulations. Gloves should always be worn when handling gels containing ETBr. The second step is setting up of the gel apparatus and separation of DNA fragments. The procedure goes as follows. The loading dye is added to the DNA samples to be separated. Gel loading dye is typically made at 6x concentration, containing about 0.25% bromophenol blue and 0.25% xynyl cyanol with 30% glycerol. Loading dye helps to track how far the DNA sample has travelled and also allows the sample to sink into the gel by increasing its density. The power supply is then programmed to the desired voltage. It is usually 1 to 5 volts per centimeter between electrodes. Enough running buffer is added to cover the surface of the gel. It is important to use the same running buffer as the one used to prepare the gel to keep the composition of the ions the same for conduction of charge. Next, the lids of the gel box are then attached to the power supply. The power supply turned on and verified that both the gel box and power supply are working. The lid is removed gently without disturbing the setup, and slowly and carefully the DNA samples are loaded into the gel. It is necessary that appropriate DNA size marker always be loaded along with the experimental samples. The cathode should be closer to the wells than the anode, as DNA molecules being negatively charged will move from the cathode to the anode. The power is then turned on. The gel is allowed to run until the dye has migrated to an appropriate distance. The next step is observing separated DNA fragments. When electrophoresis has completed, the power supply is turned off and the gel is removed from the gel box. The excess buffer is drained off from the surface of the gel and the gel is placed on the gel tray with paper towels to absorb any extra running buffer. The gel is then exposed to UV light. This is most commonly done using a gel documentation system. DNA bands show up as orange fluorescent bands. The banding patterns are to be documented with the help of a photograph that can be later used to interpret the bands. The disposal of the gel and the running buffer should be done as per the institution regulations. Southern blotting was named after Edward M. Southern, who developed this procedure in Edinburgh University in the 1970s. This technique basically involves the transfer of DNA molecules from an agarose gel onto a membrane. Southern blotting is designed to locate a particular sequence of DNA within a complex mixture. For example, Southern blotting could be used to locate a particular gene within an entire genome. The amount of DNA for this technique is dependent on the size and specific activity of the probe. Short probes tend to be more specific. Under optimal conditions, it is possible to detect 0.1 picograms of the DNA for which the probes have been made. Let us look at the technique in detail. The DNA is first digested with an appropriate restriction enzyme. This digest is run on an agarose gel. The DNA is then denatured. Denaturation is the conversion of a double-stranded DNA molecule to a single-stranded one by breaking the hydrogen bonds. This is usually done while the DNA is still on the gel. Several techniques may be used to achieve denaturation. For example, soaking the gel containing DNA in about 0.5 molars of NaOH would separate the double-stranded DNA into single-stranded DNA. This is done because only single-stranded DNA can transfer onto the nitrocellulose membrane in the next step. Fragments greater than 15 kilobase pairs are hard to transfer to the blotting membrane. A depurination step may also be done. Depurination with HCl, that is about 0.2 molar HCl for 15 minutes, takes the purines out, cutting the DNA into smaller fragments. It is necessary to note that the procedure may also be hampered by fragments that are too small. After the treatment with NaOH or HCl, it is important to neutralize the DNA with an acid or a base respectively. The denatured DNA is then transferred to the membrane. Traditionally, a nitrocellulose membrane is used, although nylon or a positively charged nylon membrane may also be used. Nitrocellulose typically has a binding capacity of about 100 micrograms per centimeter, while nylon has a binding capacity of about 500 micrograms per centimeter. Many scientists feel that nylon is better since it binds more and is less fragile. Transfer is usually done by capillary action, which takes several hours. Capillary action transfer draws the buffer up by capillary action through the gel and into the membrane, which will bind single-stranded DNA. For the transfer to occur successfully, the membrane over the gel is stacked with weights such as books so that proper contact happens between the gel and the membrane. Just as the blotting of wet ink on a dry paper transfers a replica of the image to the paper, the blotting of DNA to a nylon membrane preserves the spatial arrangement of the DNA fragments that existed after electrophoresis. A hybridization probe is a fragment of DNA or RNA of variable length, usually between 100 to 1000 bases long, which can be radioactively labeled or fluorescently labeled. It can then be used in DNA or RNA samples to detect the presence of nucleotide sequences, that is the DNA target, that are complementary to the sequence in the probe. The probe thereby hybridizes to single-stranded nucleic acid, DNA or RNA, whose base sequence allows probe target base pairing due to complementarity between the probe and the target. After the transfer of single-stranded DNA onto the membrane is allowed to react with the labeled probes against the target sequence. During this time, single-stranded DNA couples with the complementary probes. The unbound probes are then washed with a buffer. A single locus probe is a DNA or RNA sequence that is able to hybridize, that is form a DNA-DNA or DNA-RNA duplex with DNA from a specific restriction fragment on the southern blot. Duplex formation depends on complementary base pairing between the DNA on the southern blot and the probe sequence. Single locus probes are usually tagged with a radioactive label for easy detection and are chosen to detect one polymorphic genetic locus on a single human chromosome. The southern blot is incubated in a solution containing a radioactive, the southern blot is incubated in a solution containing a radioactive single locus probe under conditions of temperature and salt concentration that favor hybridization. After hybridization, the unbound probe is washed away so that the only radioactivity remaining bound to the nylon membrane is associated with the DNA of the targeted locus. The locations of radioactive probe hybridization on the southern blot are detected by autoradiography. In this technique, the washed nylon membrane is placed next to a sheet of X-ray film in a light, tight container. The X-ray film records the locations of radioactive decay. After exposure and development of the X-ray film, the resulting record of the southern hybridization is termed as an autoradiograph or autorad for short. An autoradiograph is an image on an X-ray film or nuclear emulsion produced by the pattern of decay emulsions such as beta particles or gamma rays from a distribution of a radioactive substance. Alternatively, the autoradiograph is also available as a digital image, that is known as digital autoradiography. Due to the recent development of scintillation gas detectors or rare earth phosphor imaging systems, the film or emulsion is opposed to the labeled tissue section to obtain the radiograph, also called as an autoradiogram. The auto prefix indicates that the radioactive substance is within the sample as distinguished from the case of historadiography or microradiography, in which the sample is X-rayed using an external source. In a typical forensics DNA analysis, DNA polymorphisms on several different chromosomes are characterized. After an autorad has been developed for the first single locus probe, the radioactivity on the southern blot can be washed away with a high temperature solution, leaving the DNA in place. The southern blot can be hybridized with a second radioactive single locus probe, and by repetition of the above steps, a series of different single locus probes can be seen. The set of autorads from a southern blot is known as a DNA profile. DNA fingerprint analysis is based on this southern hybridization technique. DNA fingerprinting, also termed as DNA profile analysis, is based on the use of the sudden hybridization technique to analyze polymorphic regions of human DNA. Polymorphisms are explained in more detail later on. The experimental steps used in a forensics laboratory for DNA profile analysis have already been discussed. Let us first look at the benefits of DNA fingerprinting. DNA profiling is an easy and painless method for the subject being tested. It is less invasive than taking a blood sample. The second advantage is that it is an affordable and reliable technique. It can be conducted in a relatively short amount of time, and anyone at any age can be tested with this method without any major concerns. There is also a large variety of uses, such as in legal claims, missing person cases, identification for the military, and paternity and prenatal testing. The technique has been used since 1984, making it highly developed and improved recently. The disadvantages of DNA fingerprinting are that the sample of DNA can easily be ruined during the process of DNA fingerprinting, causing the sample to become completely useless for testing. The process itself is complex and tedious and can give results that may be hard to interpret. The test needs to be run on multiple samples and a numerous amount of times for ideal accuracy. Commonly, laboratories run each test at least twice with 4 samples. Privacy issues could occur if the information isn't kept secure at the laboratory. Personal information legally can only be released with a written order. This personal information, if leaked, could potentially complicate insurance processes, healthcare, and job prospects for an individual. DNA fingerprinting can be utilized in a number of cases, such as individual profiling for suspects in homicides and other violent crimes, paternity testing, mass disaster cases, as well as non-human DNA testing, especially in wildlife forensics. Let's look at one case study. Case 1. A 15-year-old schoolgirl, Linda Mann, was abducted in Narborough, England. The next day, her body was discovered, raped and murdered. 3 years later, another young woman met the same fate near Linda's resting place. Richard Buckland was arrested and confessed to the second murder only. An untested technique was applied that was known as genetic fingerprinting through DNA analysis. Surprisingly, there were no matches in either murder, so the test was repeated. Ultimately, Buckland was proven innocent. As to why he confessed, he claimed that he had been pressured by the police. Now, 5,500 men from the local area were then tested. Colin Pitchfork persuaded a friend to test in his place. But when he bragged about fooling the investigators, he was overheard and reported. His genetic profile matched the semen samples from both the girls, and in 1987, he became the first murderer to be convicted by DNA analysis. Case 2. One of the most famous cases in India involving the use of DNA fingerprinting is of N.D. Tiwari. N.D. Tiwari, a famous politician, was alleged by Rohit Shekhar to be his biological father. In the subsequent paternity case, many legal issues surrounding the DNA testing emerged, such as if paternity is conclusively proved, that is 100% by DNA testing, whether the persons have the right to keep the result of such tests private, or so on. Indian law says that one cannot be forced against his or her will to give DNA evidence. But today, in certain cases, courts are forcing suspects to undergo DNA test. As in the N.D. Tiwari case, all these arguments were also raised, but the court forced N.D. Tiwari to give his blood sample in the larger interest of the public. In the larger interest of the public. Courts can now force people to give their blood samples to meet the demands of justice. As such, the paternity of the alleged father was thus proved with the help of DNA fingerprinting. In conclusion, DNA fingerprinting or profiling now forms the basis of a plethora of forensic investigation protocols. It is essential to understand these techniques involved in the It is essential to understand the techniques involved in this DNA profiling to allow better utilization of DNA. DNA profiling relies heavily on databases. As such, it is essential to form extensive DNA databases for humans as well as non-human sources. Only then can the use of DNA analysis be exploited to its full potential. Although India is currently lagging in this area due to insufficient databases, DNA analysis still has taken a firm root in the country as a major tool of forensic investigation. With this, we come to the end of today's chapter. I will return back with a new set of chapters for your learning and understanding. Till then, thank you and goodbye.